Okay, let's pick up where we left off, shall we? So we're talking about the protein filaments of the sarcomere, which is like the contractile. Yes, good. Okay. The contractile component. A little bit about the actin subunits, the proteins that are involved in the actin chain. And now we're looking at the myosin. So the myosin is basically kind of a long shaft-like protein. It's thicker in diameter than the actin, and so it has essentially a shaft-like portion of it called the rod. And then at the tip of it, it actually has kind of like a hinge and then a little head group. So there's like a hinge and then the actual myosin head. Now, the myosin heads are important. So the head, I just wrote one of them because the other one is kind of behind it. So there are two lateral heads, they're kind of like that. And you have your hinge piece here. So you actually have two heads. And so what these guys are going to be doing is creating cross bridges with the actin molecules. And so when they bind to the actin, they basically are going through the same mechanism as a motor protein would go through. Now, the important thing to understand about a motor protein is an important class of proteins. Generally, you would learn about this in general bio, but sometimes some instructors don't talk about it. But they are a critical type of protein because they're the ones involved in all locomotion. So like whether or not you're moving some sort of a vesicle around inside the cell, flagellar motion, cilial motion, these are all basically driven by motor proteins. When you go through mitosis, when the DNA is moving from one end to the other, it's all being moved by motor proteins. Pretty much if anything's moving in the cell, it's a motor protein that's moving it. There isn't such thing as random drift in the cell. The cell doesn't like random. Random is expensive and difficult. But here's basically how a motor protein works. A motor protein will have basically a couple of major domains. So it'll have, and typically what they do is they essentially walk along microtubules. So a microtubule is essentially like the track on which your motor proteins will walk. And so generally speaking, you have a walking domain, and then you have a cargo binding domain. And generally, like if you're moving a vesicle, you'll have essentially the cargo being moved as it's being bound by the cargo binding domain. Now, the way that you get this to work is basically it requires energy. So ultimately, you're going to need some ATP, which is actually going to come in here. It's going to hydrolyze, and it's going to give you ADP plus phosphate plus energy, and that energy is going to be used to actually walk. And so it goes through a similar type of mechanism of walking. And so what they do is they essentially grab onto stuff and they essentially walk, just the same way you walk, down this microtubule structure. So they're involved in all kinds of movement. And the reason why we mention that is because myosin is essentially a type of motor protein in the sense that what they do is they attach and then they pull, right? So like when you're walking, you have the exact same mechanism that a motor protein has, right? You walk in a cycle. So for instance, when you put your foot on the ground, that's the attachment, right? That's the first step of a motor protein doing its walk. Then once your foot is attached to the ground securely, then you have a power stroke. You pull yourself along by the power of your leg and you pull yourself forward. So that's your power stroke. And then after that, you have a detach. So you pull your foot off of the ground, only to do it all over again. You basically swing your foot forward, you attach, power stroke, detach. And then you pull your foot forward, attach, power stroke, detach. And that's the exact same thing that the motor proteins are doing. They need ATP to do that. By the way, you also need ATP to do that. So to walk, right? So you need energy to be able to walk as well. So these are basically motor proteins. In terms of a motor protein, what would be similar to the walking domain, that is the part that attaches, pulls, and then detaches, is going to be the head group. So this is like the walking domain. The rod is similar to the cargo binding domain. But in this case, it's tethered to a stationary point. So when the myosin heads pull, you don't walk your myosin forward because your rod is attached to something solid. What ends up happening is instead of pulling the myosin forward, what happens is you end up pulling whatever your heads are attached to forward or backwards. Okay. Just the same way as you have the physics of a tug of war rope. When you grab onto the tug of war rope and you pull it, there's two different outcomes that could happen. Number one, if the rope is stationary and attached to something that will not move, what's going to happen is you are going to pull yourself forward, right? The second outcome is if you are the one who's attached to the solid substrate that will not move, then you will pull the rope towards you. And so you see both conventions happening when you're talking about motor proteins. In this particular case, it's the pulling the rope towards you scenario because you are tethering yourself to the hard surface of the sarcomere. So ultimately what has to happen is your myosin heads have to make contact with the actin, has to grab onto it, similar to you having to basically grab onto the tug of war rope. You have to make contact with it. When you do, and you bind to the actin molecules, you form what's called a cross bridge. Now, once you're attached to this rod portion, then what can happen is the hinge, which can straighten during contraction, is then going to allow the head group to essentially pull on that actin. But it does so with ATP. So ATPase, which is an enzyme, will basically break ATP into ADP and phosphate, but it'll release that energy. And so some of that energy is going to be used to bend the hinge region of the myosin, and then that will pull on the actin. Just like when your hands are grabbing onto the rope, energy will allow your muscles to contract and you will bend your hinge, in this case, like your wrist, and you'll pull the rope toward you. And this is kind of what it looks like in terms of the major fibers. So once we actually clear both of these thin and thick fibers, then we'll start taking a look at both of them together. So as a visual, because we haven't seen this yet, and to kind of go back a little bit and to review in terms of the actin filament, the thin filament, this is basically what it looks like. So you can see the purple circles are the actin fibers. Remember, we have fibrous actin, that is the F actin. Oops, sorry. You have the fibrous actin, which is the F actin. And then the G actin, if you see, notice here, is going to have these actin sites, these little black sites here. I'm going to color them red so you can see they stand out a little bit better. So these are little binding sites for the myosin heads. In addition to that, you can see your tropomyosin, which is this long rope-like protein. Typically, what's going to happen is this tropomyosin, as it weaves through this, is going to cover up those actin sites and basically make them inaccessible. So that normally you cannot form cross bridges. Now, if you take a look then, attached to the tropomyosin is going to be your three-part troponin. Remember, troponin has a couple of binding sites. One is for tropomyosin, so it's going to bind to tropomyosin on one point. On another piece, it's going to bind to the actin. And then the third one is going to be the calcium binding area. And so you're going to have these basic ones. So they kind of have it positioned. They have it in a particular format right now. But you can see you have these three different pieces. And so they have these backwards. I actually have these backwards. So calcium and G-actin. So this isn't actually an image that's kind of true to form. It's just kind of showing you the three binding sites. But basically, the G-actin one is bound and to the actin filament, and then the calcium one is kind of waiting for calcium to come by. And so that's basically your default state. So normally what happens is the tropomyosin is covering these G-actin sites. Troponin is attached to the tropomyosin and the actin filament and waiting for calcium. So that's basically kind of where you are. That's where your setting is at rest. Now, here's the question. What we want to do is we want to know, how do you get the muscle to contract? Or the sarcomere to shorten, which basically will lead to muscle contraction. Well, first of all, you got to get a signal, right? You got to get some sort of a communication from a motor neuron that's going to basically tell you, okay, time to contract. So what we have to do is we have to take a look, first of all, at the front end. So the sarcomere is kind of like the back end of the muscle response, where you're actually doing the contraction, the physical contraction. But now what we want to do is we want to kind of back it up a little bit. So we kind of, if you just to kind of follow along, and what I want to just kind of highlight, it seems a little disjointed, I know, but what we're doing here is we're basically covering different pieces of a very long continuum, right? So we started kind of in the middle, talking about the structure of the T tubules and the terminal cisterna and the triad. So that's one piece, the middle piece. Then we kind of skipped forward to the structure of the sarcomere and how the sarcomere can shorten, and that is kind of the end piece. And now we're going to be going back to the beginning and taking a look at how we kind of get the beginning piece put into place. And then once we get all the pieces sort of sketched out, then what we're going to do is we're going to tie them all together in one long continuous process, and we'll kind of pull it all together and connect all the dots. That's basically what we're going for. Okay, so just in case it seems a little disjointed and kind of haphazard. So where we are right now is we're basically jumping back to the beginning to figure out exactly how do we get the actual contraction signal into the muscle so that we can actually get muscle contraction. And so the first thing we need to do is take a look at the motor neurons, right? So essentially the motor neurons are going to be the ones responsible for carrying the electrical signal. This, by the way, if you're a muscle, is the contraction signal. So this is what's going to stimulate you to contract. And the signal is going to be carried by what's called an action potential. We'll talk more about that in the neurophysiology chapter in chapter 11. This will stimulate the muscle fibers, who will then relay their own action potentials until ultimately that terminates into shortening of the sarcomere. So first of all, we need to talk a little bit about the interface between the motor neuron and the muscle fiber. That interface we call the neuromuscular junction, or for short, the NMJ. So the neuromuscular junction is basically synonymous with the term synapse or neural synapse, and some of you may be familiar with that term. And so the pieces of the NMJ that we have, first of all, begin on the neuronal side. So the first place we start is what's called the presynaptic terminal. So presynaptic, before the synapse, terminal. So this is the axon terminus. So essentially a action potential has been galloping down the axon, heading toward its target. In this case, the target is a muscle because we're talking about a motor neuron. And it arrives now at the presynaptic terminal. And waiting at this preaxonal terminus are a series of what's called synaptic vesicles. And so inside these synaptic vesicles are neurotransmitters. And in this particular case, when you're talking about a motor response, that specific neurotransmitter is acetylcholine, or ACH for short. So basically what you have at the axon terminus is basically these synaptic vesicles. So you can see them here. That are full of acetylcholine. Now, what happens is as the action potential goes down, so here's kind of what it looks like. We're blowing this image up. As the action potential goes through the neurolemma of the axon, then it's going to arrive at the axon terminus where these synaptic vesicles await, loaded up with their acetylcholine neurotransmitter. So ultimately, when you have a neurotransmitter, this is essentially a chemical that can relay the action potential across the synapse to the target cell. So essentially, the neurotransmitters will be released, and on the other side, they can bind to ligand-gated ion channels of another cell. Now, that's a mouthful, right? Ligand-gated ion channels. We'll talk more about that mechanism here in just a little bit. But the space that we're talking about, the synapse, is the synaptic cleft. That's essentially what has to be breached. So between the neuron and the target cell, in this case, the muscle, is a gap. This gap is your synaptic cleft. So you have to basically get your message, your action potential, across that gap, that space, and received by the target cell, the muscle, so that you can essentially reignite the message to the actual muscle itself. So there's a bit of a transmission idea that's going on here. Now then, on the other side of the synaptic cleft, or the synapse, is the postsynaptic membrane. So this is basically the membrane, the plasma membrane of the target cell. And sometimes we refer to it as the motor end plate. So sometimes you'll hear that oftentimes in muscular physiology. And so this is basically the structure of what we're trying to do. So we have to basically jump this gap. That's essentially what we're doing. Now, a couple of things, first of all, and what I want to do, hold on one second. Let's see if I put that slide in here. No. Okay. So I want to blow this up just a wee tad so you can see it a little bit better and kind of finish my thought over here, and hopefully it doesn't get too poofy. What happens is as your action potential ripples down the neurolemma, what's going to happen is it's going to come to a series of channels. In this particular case, these are ion-gated channels. And this will basically open up under the stimulus of this action potential, this electrical signal that produces a voltage change. And so what's going to happen is this is going to allow these ions to flood inward following their diffusional gradient. And that is the signal or synaptic release. What release? For these vesicles to go to the presynaptic membrane and release their acetylcholine. So you can see you have here a two-part molecule, acetate and choline. And then on the other side, you have these ligand-gated receptors, ion channels, waiting for the acetylcholine. So what's going to happen is acetylcholine is going to bind to these gates. It's going to open them up, and that's going to allow sodium to rush in, and that's going to re-trigger your action potential, and that's going to start galloping down the sarcolemma. And so ultimately then what's going to happen is you're going to get the release ultimately of calcium as this action potential dives down the T tubules and opens up the sarcoplasmic reticulum. So what I want to do is I want to kind of go back and forth a little bit so that we can see some of these images. Some of these are recapitulations of what we saw before. For instance, we already saw the cross-sectional view of the myosin filament. We already saw the troponin and tropomyosin of the actin filament. When you put these two guys together, you'll notice that what you have is a region of overlap here where your myosin heads will attach to the actin and then pull in this direction, pulling your Z discs inward, shortening your sarcomere. That's the sliding filament idea. So we'll take a look at that more closely. What I want to do is I want to come back to this image. I want to kind of jump back and forth a little bit, so just bear with me, because there's some things that I want to take a look at, and I want to sort of sketch out a little bit and then kind of come back to this and start filling this in a little bit with a bit more detail. The first thing we want to do is we want to take a look at this sort of sliding filament concept, right? So the idea of the sliding filament model is the idea that the actin myofilaments are essentially going to be sliding over the myosin short filaments in the sarcomere. The actin and the myosin themselves do not change length. They just slide relative to each other. So for instance, when you have your Z disc here, you have your actin filaments, which are thin, attached to your Z disc like this. Then in between, you're going to have your thicker whoa, that's really thick. Okay. Not quite as don't want it quite that thick. I'll just there's your myosin, and in your heads are going to be attached basically to the actin. Now remember, the myosin is in 3 dimensions all around attached to the actin. And so what's going to happen when these heads pivot, they're going to basically pull in this direction. And so they're essentially going to pull the actin in toward each other, and that's going to shorten the sarcomere. Because if you recall, the head of the myosin is going to pivot in this direction under ATP. So it's going to basically go from this relaxed state, it's going to attach, and then it's going to pivot the power stroke, and it's going to basically pull forward. It's going to pull that actin forward. So it's going to go like that. And so when you do that, you're basically going to be pulling your actin in toward each other, and so they shorten. And this is basically if you shorten your sarcomeres, which they will shorten across all the sarcomeres, then you're going to shorten your muscle and you're going to create tension. And this is muscle contraction. During relaxation, muscle relaxation, your sarcomeres will lengthen because oftentimes there's an opposing set of muscles which will then pull the sarcomeres apart. And then that'll kind of stretch out your sarcomeres so that you can contract them again. So one of the things I want to take a look at here is in sarcomeric shortening. So here you can see a relaxed sarcomere. You can see you've got a healthy H zone. So you have a good zone of overlap. So all of your myosin heads are attached now to your actin. And so once they pull, they're going to contract. Notice how the myosin heads have basically pulled away from the Z disc, pulling those actin chains in toward the centerpiece. Notice how your H zone disappears. That's because the actin is moving and sliding in and filling in that H zone because they're getting closer together. You can still see the M line, but you no longer see that nice white H zone. Notice also you can see that you've got a noticeable shortening of the sarcomere. So here's one sarcomere from one Z disc to the other. And now you can see you've got a shorter distance between Z discs. So the sarcomere is effectively shortened as the myosin has pulled on those actin chains and pulled them toward the center, pulling the sarcomere inward. And so when you do that across multiple sarcomeres, it's going to shorten the muscle, and that muscle is going to create tension. Now, one of the important things is this contraction mechanism is going to be stimulated by the action potential that jumps over this synaptic cleft. And once it's received by the receiving ligand-gated ion channels in the sarcolemma, then you'll reignite the action potential in the sarcolemma, and then it'll basically go down into the T tubules. Okay. So the nervous system is what controls muscle contraction, especially skeletal muscle contraction, and it controls it through action potentials. Now, here's the one thing to understand, is that when you're taking a look at the membrane, remember the membrane, when you take a look at a cell membrane, what you see in a typical neuron in particular, and of course the muscle, is essentially you see a gradient of sodium and potassium that's created by the sodium-potassium pump. Generally speaking, the sodium-potassium pump will pump 3 sodiums to the outside of the cell. But at the same time, because it's an antiporter, it'll pump 2 potassiums to the inside of the cell. So what you're going to create now is you're going to have a membrane. So here's my membrane. Your sodium-potassium pump, which is the poster boy for all pumps, is going to take 3 sodiums. It's going to pump it to the outside. And it's going to take 2 potassiums from the outside and pump it to the inside. So what's going to happen? So ultimately what's going to happen is as you add 3 sodiums to the outside relative to 2 potassiums going inside, you're slowly going to build up a concentration gradient where sodium is higher on the outside and lower on the inside. You're also at the same time going to build a potassium gradient where you have lower concentrations of potassium on the outside and higher concentrations of potassium on the inside. So you have 2 different concentration gradients that you're building here in opposite directions, with a slight favoring in the strength and the size of the sodium gradient. So you're going to have a little bit more sodium on the outside than you have potassium on the inside. Now, the reason why I want to sort of explain that and sketch that out is because what happens is when you're pumping 3 out and 2 in, remember, you're not just pumping sodium or potassium. Sodium is an ion, as is potassium. But they are positive ions. So what that means is for every pump of the sodium potassium pump, you are actually building up a slight positive charge on the outside. It's looking a little on the positive-ish side because you're building up one extra positive charge on the outside relative to the inside, which is not necessarily negative, but relatively it looks less positive. So it kind of looks negative. Now, here's the thing. Whenever you have charge separation or charge disparity separated by an insulator, in this case, the membrane is an insulator, you get a voltage. That voltage is basically the resting membrane potential. This is the membrane voltage across the membrane that's attributable to the pumping of sodium potassium pump. We can actually measure this voltage in muscles. It actually comes in to be about minus 70 millivolts on average. But what that basically means is you have in your cells the physics, the electrical physics, that is exactly the same thing as what you get with static electric charge. Like for instance, when you shuffle yourself across a carpet and you're building up electrons on yourself, and you go and you reach that metal doorknob, what happens to you?
You get shocked.
Yeah, you do. I like your zap, Daniel. Just very succinct and very appropriate, right? So what is the zap? What the zap is actually the flow of electrons. It's basically electricity. And that's what all static phenomena is. It's basically two areas with a disparity in charge. And it gets so big that eventually what happens is the insulatory capacity of what's separating it. In your case, it's air that's insulating you from the doorknob. But at some point, what's going to happen is there's not going to be enough insulation between you and the doorknob to keep you insulated. And what happens is essentially your electrons flow from you to the doorknob, and you feel that as a static shock. By the way, this is the same physics as well that exists in lightning. Lightning is simply just static charge and static electricity on a grand scale. Basically, two clouds rub against each other, rubbing electrons from one to another. One accumulates electrons, the other one looks relatively electron deficient, and air is insulating them. And when that disparity builds up to a great enough capacity, it'll actually break down the air. There's actually a physics measure called the dielectric. It's called the dielectric effect. And there's actually a constant for air for different substances. And so once it exceeds that dielectric ability for air to insulate between those two, then you get a static arc basically between the two clouds. And we see that on a grand scale as lightning. So lightning, static shock, and your physics, your physical physiology, your membrane potential are all essentially the same set of physics. So the reason why I mention this is because it's going to be very important. In order to understand what's going on in this image, you're going to have to understand some background physiology associated with what's going on in some of these channels and in some of these neurons. We're going to have to talk neurophysiology before we get to neurophysiology. So just act surprised once we get to chapter 11. So ultimately, this has to occur in order to get an action potential. So your sodium and your potassium drives your action potential. So ultimately then your phospholipid bilayer, because it is hydrophobic and it does not allow passage of ions, right, particularly positively charged ions and negatively charged ions, this is what creates and gives it this sort of electrical property as an insulator. By the way, as you guys get to AMP2, you're going to notice that oftentimes what happens when you're moving charge around and your goal is to not build up charge, like voltage, like membrane potential, oftentimes you have to balance your charge. Like for instance, if I didn't want to create a membrane potential physiologically, every time I moved a sodium, I would have to balance that out by either neutralizing it with a negative charge, by moving a negative charge along with the sodium and essentially neutralizing the charge differential, or by taking an equal positive charge away from the other side and moving it in away from it. So basically kind of eliminating positive charge to kind of keep it all the same. So when you get deeper into physiology, you'll see there's a lot of charge balancing techniques and mechanisms in physiology in scenarios where you're not interested in building up a membrane potential. In this case, we are. And so we want the membrane potential to exist because that's going to be the basis of our ability to send an electrical impulse. And so then ultimately, once we build this electrical potential, then our transport proteins, especially our channels, can basically play a role in creating this kind of electrical voltage change as we allow these positive ions to basically move through. Now, a couple of things I want to cover and I want to go over. What I want to do is there's a couple of things because we're talking a little bit about control and regulation through these channels. And there is channels basically are associated with diffusion, facilitated diffusion, right? So basically you've got some sort of a molecule that's going to be moving from an area of high concentration to an area of low concentration. And in this case, it's usually of a polar molecule like water, or a charged molecule, which is hydrophilic like sodium. And you basically need to create safe passage so that it can get through this hydrophobic nonpolar membrane. And basically there's different ways that you can control ion channels. And as a matter of fact, almost all of them, all four techniques are essentially physiological in terms of how they're broken down. And so what I want to do is I want to insert a slide and I want to put them all in one place. We've got two of them on this slide, but what I want to do is I want to put the other two in the same place so you kind of have them all in the same area. Okay, so this is a slide that I affectionately call channelology. So this is the control of movement through a channel. Now, once I get done with this slide, we're going to have the basic understanding to be able to go back to this particular slide and to understand it in better detail. So what we're looking for is how do you control how a molecule moves through a channel? There's four major ways to control this. One is with what I refer to as osmotic control. This is basically where you control the flow of a molecule with osmotic forces. So here's an example. So I'm going to draw a membrane, and then I'm going to have a channel. And I'll put another channel in there. And so in this membrane, this is the outside, this is the inside. And not only that, but along with this, I have a gradient. So I've got a high sodium gradient out here and a low sodium gradient on the inside, just like our electrical potential, our membrane potential. So now if these channels are open, which direction, as I've got it set up now, which direction is the sodium going to flow? Answers. Ooh, I'm making your heads hurt, aren't I? This will require you to revisit your understanding of diffusion and osmosis. So let's start there first. Somebody define diffusion for me. This is just like general bio review. I can hear some of you out there Googling it right now. Yes, thank you, Ashley. So a flow from an area of high concentration to low concentration. And specifically, when you take a look at diffusion, the flow that we're talking about is the flow of solutes, right? So it's the movement of solutes from an area of high concentration to low concentration. So given the definition of diffusion, and from what I wrote here on the screen, which direction would you expect sodium to flow? Yes, it's going to go from high to low. So it's going to flow into the cell. Notice, how did I get it to flow into the cell? Simple. I just basically set up its concentration gradient in such a way that caused it to flow into the cell. What if I wanted it to flow outside of the cell? I just turned the concentration gradient around. I put the high concentration on the inside and the low on the outside, and it's going to go out of the cell. So this is a way to control that. By the way, osmotically, what's going to happen? Which direction is water going to flow? How do you define osmosis? It is indeed the diffusion of water across the membrane. But the thing is, when you're looking at osmosis, there's a mantra to osmosis to understand which direction water will move, right? So you go from low to high. So you're talking about solutes, right? So in osmosis, we have a mantra: salt sucks. So basically, where the salt concentration is going to have a sucking effect on the water. Another way you can look at it is water chases salt, right? So in this particular scenario, which direction is the water going to go, into or out of the cell? Yep, it's going to go out, right? Because that's where the salt concentration is. And by osmosis, it's just basically going to go out and it's going to chase that salt pile that's outside. So what have I done now? What I've done is I've controlled movement across my membrane through my channel simply by manipulating these osmotic pressures, right? Knowing that water chases salt and that salt wants to flow down its gradient, I can utilize that to dictate how these things are moving back and forth across the channel. By the way, this is the primary mechanism that you use to move water in your body, is by utilizing osmosis and using this kind of osmotic control situation. The other one and the other three that we have, I want to cover because it's going to allow us to understand the nature of our neuromuscular junction. The next way to control movement through a channel is through our ligand-gated channel. So basically, this is what it looks like. In this particular case, you're going to have your membrane. I'm going to have my channel. And in this case, I'm going to have a gate. It's a protein that has a special little pocket on it that is going to bind to a particular signal molecule, a ligand. So this is basically a ligand, which is a chemical signal. And so when this chemical signal binds to the gate, what's going to happen? It's going to open up the gate. Now, the same set of conditions is true for this one as well. So when that ligand opens the gate, what's going to happen to the sodium? Where's the sodium going to go? So by diffusion, it is going to move from high to low. So it will rush in. Notice though, you've controlled the flow through that channel. That's not going to flow from high to low unless you open up that gate. And guess who has control of that chemical ligand? You do. So you get to determine how to open that gate and when to open that gate. Okay. So that's a ligand gated channel. The other one is a voltage gated channel. And I'm going to talk about that one here in just a second, but I want to end with that one. So what I want to do first is I want to talk about the fourth one first, because it's actually in our senses, and we will take a look at that one. But I want to do that one first, and then I want to finish off with the voltage gated, because the voltage gate kind of has a way of tying everything together. The fourth one is a mechanical gated system. So in this case, basically use the physical distortion of a protein to actually physically open your gate. So here's a good example. I'm just going to draw the pictures right now, because pictures are more instructive right now than me writing a bunch of words that you guys can't even read anyway. So here is your gate. And typically, along with this gate is a protein that has distortable ability. So this is a distortable protein. And this is attached to a hinge protein that is tethered to the gate. So now what happens is if you were to distort this protein and basically move it in this direction, then it'll flop over here. And as it does, it's going to pull the hinge, and that's going to pop the gate open. And then, of course, you have the same set of conditions here. So you have your high and your low. And so when that pops open, then what's going to happen is your sodium is going to rush in, just like the other two scenarios, right? Now, it's a mechanical gated system. Matter of fact, this is actually how you hear. So one of the things that we'll see, and I'll just go ahead and lay it out now, just because, I mean, it's good physiology, especially for our senses. But basically, the way that you hear is that compression waves in the air. So sound is actually a series of waves. So your sound actually compresses the airwaves into peaks and troughs that have various wavelengths to it. They're either very short or high frequency, or they're very sort of long and low frequency. And so what happens is these airwaves will hit your eardrum, your tympanic membrane, which will vibrate back and forth. Now, in your middle ear are your auditory ossicles, right? The so-called hammer, stirrup, and anvil, right? And so those are attached basically to your eardrum on the outside, and they're like a hinge structure. So basically, as your eardrum vibrates, it basically pulls back and forth on those auditory ossicles. On the other side of your middle ear is another membrane called the oval window. And this is kind of like a thin physiological membrane that your ossicles are attached to. And so as the airwaves vibrate your eardrum, your auditory ossicles will move back and forth, and then they will pull back and forth on your oval window membrane, kind of like a wave machine. On the other side of the oval window is your inner ear. And in your inner ear is fluid called endolymph and perilymph, but it's a fluid that goes in your inner ear. And so as your oval window vibrates, it creates waves and recapitulate those waves in the fluid in your inner ear. Those waves will flow over a distortable membrane that will then basically open your mechanically gated sodium channels. If your gate opens just a little bit because the waves are gentle and not severe, then it'll only let a little bit of sodium in and you hear a particular pitch. If the waves are strong, then the distortion is greater and it'll open up that gate much wider and you'll get a much stronger inrush of sodium and you hear that as a different pitch. So basically, your ability to determine high pitch, low pitch is essentially a mechanical gated system. That's essentially the basic mechanism of how you actually hear. Now then, the good one, the voltage gated channel. Let's take a look at this one. So in the voltage gated channel, what you have are a series of channels that are stimulated to open when the voltage changes. So let's go ahead and take a look at this. So here's a series of channels. So these guys are all going to be voltage gated. I'm going to put a big V right there. And I'm going to start it off with a ligand gate. So now let me set the stage, right? Remember, on the outside, we have a high sodium concentration. On the inside, we have a low sodium concentration. Because of that, the disparity of positive charge, basically, it gives us a membrane potential. So it looks slightly positive on the outside, slightly negative on the inside, creating a voltage. That's your membrane potential. Now the voltage gates basically are stimulated to open when they sense a change in voltage or membrane potential. So let's go ahead and take a look at this. And the reason why I wanted to start out with a ligand gate is because that's exactly what we're going to be taking a look at. So imagine that the first thing that happens is a ligand comes in here, binds to our ligand gate, and then basically pops that gate open. What's going to happen to the sodium as soon as that ligand gate opens up? So we can go off of Daniel's previous answer into the cell, right? So it's going to basically rush into the cell. But remember, in this particular case, remember, it's not just sodium that's rushing into the cell. What's rushing into the cell? Positive charge. So what's happening is all that positive charge that was on the outside is now rushing into the inside. As a result, you get a charge flip. Now all of a sudden the inside looks positive relative to the outside. And you basically flip the membrane charge. So it starts to look negative on the inside or on the outside instead of positive. That's a voltage change. It's what's called depolarization. And so once that voltage flip is sensed by the voltage gate, it will pop the voltage gate open, which does what? It allows more sodium to rush in. So you get another wave of positive charge, which basically will depolarize the next membrane and flip that charge. That voltage change will then trip the next voltage gate to pop open. And what's going to happen? In rushes, again, your sodium, and you have a positive charge in rush. That's going to flip the adjacent membrane. That's depolarizing adjacent membrane, causing a voltage to trip the next gate, and then so on and so forth until you get this sort of like domino-like effect of depolarization. So basically, as soon as you tip one gate over, then what you get is this sort of rolling domino-like succession of voltage gates that are opening up. That is basically how you propagate an action potential. So with that, then let's take a look at this. So when that action potential comes in, what's going to happen is it's going to trigger voltage sensitive calcium channels, which are going to allow calcium to inrush, and that's the signal for these synaptic vesicles to release, in this case, acetylcholine. But what is acetylcholine? Acetylcholine is a neurotransmitter. It's a ligand, and it's going to open up these ligand-gated sodium channels. So not only are they ligand-gated, but these are acetylcholine-gated sodium channels. So as soon as the acetylcholine binds to those channels, it opens them up, inrushes the sodium, that creates a depolarization event, and that depolarizes the adjacent membrane. Now what's going to happen is it's going to basically go down and it's going to start tripping these voltage gated channels that are in the sarcolemma. So as this depolarization moves toward the voltage gates, it'll start to trip one domino after another, after another, after another, after another, after another, just like a line of dominoes falling over. And then now you've got this rolling depolarization, this voltage flipping that's going racing through the sarcolemma, and then it hits the T-tubule and it dives down into the shaft like T-tubule on its way to the terminal cisterna. Okay. By a show of thumbs up or smiley faces, how much sense does that make? Or I guess you could do frowny faces too if they don't make any sense at all. Okay. So that's good. That's good enough. So about 50-50 is about good enough. It takes a little bit of a process for it to kind of all sink in, but it's very sequential and it's very stepwise. So let's take a look at the resting membrane potential. Remember, this is basically at rest. And so when we take a look at the resting membrane potential, what we're going to see is a system where we have our different gates. And so we're going to have the pink ones are going to be sodium, what's called sodium leak channels. And then the purple ones are potassium leak channels. Now, a leak channel is basically a channel that conforms to our first convention. It's open, ungated, and just basically is following osmotic control. So molecules are free to sort of come and go as they wish. Now, what happens is your sodium potassium pump, which is basically building up your sodium and your potassium, is accompanied with a series of leak channels, both sodium leak channels and also potassium leak channels. And so the idea here is to maintain a stable membrane potential, right? So the idea is you don't want it to build up and get too extreme. You want it to be stable, which means you have to let some of these sodiums to sort of come back and forth so that they don't build up too aggressively and create too much charge separation. So ultimately, when you set up your gradient, because you've got a high sodium gradient on the outside of the cell, when these voltage gated channels open up, the sodium enters the cell. And so the gated potassium channels will do the same because they've also got a concentration gradient. So they will leave, these guys leave the cell. And so when you have excitable cells like neurons and things like that, they oftentimes will have lots of potassium, what's called leak channels in them. And so when you see a cell at rest, that's basically one that doesn't have an action potential going through it. It's essentially maintaining its membrane potential. Generally speaking, your potassium will move out of the cell a little faster than the sodium is moving into the cell. And so that's basically because of the negative charge of the cell inside versus outside. And so it's going to be, you're going to be moving those charges roughly in the opposite direction. And none of that, but there's a stronger impetus for the potassium to leave because of its concentration gradient. Whereas you basically regulate the movement of sodium into the cell a little bit more tightly. You don't want the sodium to just come all back in. You want it to come in under your circumstances. So ultimately, as we sort of are at rest, what you're going to see is a series of gated channels and these leak channels. So what's going to happen is as you have your sodium concentration on the outside, you will allow some sodiums to leak through and to come back in because your pump is constantly going to be pumping that back out. You'll get a little bit leaking in, but then you'll pump that back out. And so you want this to happen because what this will do is this will basically help you to maintain a kind of a constant distribution of sodium and potassium. So essentially, as these are going back and forth, so you'll see that as the sodium moves in, you'll have positive charge coming in, which will cause the positive charge of the potassium to want to move out and back in. So there's a couple of different forces that we have to keep track of here when we're talking about just movements of ions across membranes. Number one is the positive ion will be repelled by a negative environment, so it'll want to move away from that. Number two is it'll always want to move down its concentration gradient. And number three is the positive ion will be repelled by its own like charges. So for instance, if you basically get a lot of positive charge built up inside, it's going to want to repel positive charge out of it to get away from the repulsive forces of positive charge. In general, when you're looking at your leak channels, what you'll end up having roughly overall is the slow leak of sodium coming in because of its concentration gradient, and the slow leak of potassium's going out because of its concentration gradient. And so you'll kind of have these guys going back and forth and back and forth and back and forth where they'll reach equilibrium. And so the leak channels will basically allow you not to indefinitely build up charge, right? Because you only want to get it up to roughly about minus 70 millivolts. And so with your leak channels, then you add those to your powerful pump, which is preferentially putting sodium to the outside and potassium to the inside. And so that's basically what's happening here. So you essentially have sodiums coming in through leak channels. That's allowing the charge to not build up too positively. So the inflow of sodium allows your membrane potential to not get so severe. If you didn't have leak channels, then you would indefinitely build up your membrane potential and you would literally statically shock yourself at a physiological level. The amount of charge difference would just get up to such high levels that eventually it would break down the insulatory power of your membrane and it would shock you. So you'd have like little electrical arcs at a physiological level. The leak channels allows you to balance that charge and to keep it from building up too high so that you're not statically shocking yourself. And that's also, by the way, the reason why potassium is kind of wired in the opposite direction. It's the reason why as your sodiums are coming in, your potassiums are going out, and then the pump is basically counteracting that. The reason is because the potassium also helps to buffer against the extreme buildup of sodium on the outside. So potassium is like a counterbalance. It's like a counterweight to the movement of the sodium charge. Okay, so what is the moral of that story? Your sodium potassium pump is building a charge separation on the outside of your membrane. Sodium is building up and building up and building up. However, you have leak channels for potassium and for sodium. And what these leak channels do is it basically allows sodium to come back in passively, and thereby it manages to keep the buildup of charge limited on the outside so that you're not going to statically shock yourself to death. Okay. You don't want to indefinitely build up charge. And this maintains your equilibrium. This is the reason why we could say your resting membrane potential is minus 70 millivolts. Well, how come it's not more? How come your resting membrane potential doesn't just keep increasing? The way, by the way, it does when you shuffle across the carpet. The reason why you get shocked is because you don't come to equilibrium. The reason why lightning strikes is because the clouds don't come to equilibrium. They don't have an outlet for those electrons to just passively move away from them. They just keep building and building and building and building until they reach a crisis point, and then boom, lightning. But what your membranes are organized to do is to eliminate the lightning effect and to allow that balance at equilibrium so you can maintain a static membrane potential. Okay. That's a hard one to wrap your head around. How did that one come across? Is that one making sense to you guys a little bit? So now you guys are probably going to be like going from somewhat getting there to like, okay, you just totally lost me. That's okay. Spend some time with that one. The reason why I wrote these pieces down here is because these are the take home, right? The take home point is all about equilibrium, right? You're balancing that buildup of charge so that you don't get static crisis, right? Physiological electricity. You don't want physiological electricity. That's what the whole point of this slide is. Okay. Now let's go ahead and take a look at action potentials. Just want to sketch out a couple of things here. First of all, an action potential is essentially a change in voltage over time. This is basically the process of your voltage changes during a stimulation event. And you have two different phases. We already talked about depolarization, right? So depolarization is when the inside of the membrane becomes less negative because of the inrush of sodium. And that basically causes a charge flip, and that's depolarization. Repolarization is essentially when you return back to resting membrane potential. Oftentimes, it's basically a resetting of the system. Due to the sodium-potassium pump. So you close all those gates and you let the sodium-potassium pump restore order and get yourself back to resting membrane potential. So your pump is key in resetting the system. Typically speaking, when you take a look at an action potential, one of the important things to understand is that the action potential is an all or none principle. You don't have like, you know, a little bit here or a little bit there or partial potentials. You have a threshold that you get up to, but then once you get to that threshold, boom, it's like it's all or nothing. It's like boom, you get it or you don't. And so the other piece that we have to the action potential is the concept of propagation. Once you get a depolarization event, how do you propagate that? How do you spread it from one region or one location of your axon, for instance, to the next, so that you can get the thing to gallop toward your targeted cell? And then the other piece you have with action potentials is the frequency of action potentials, or the number of stimulations per minute or per second. And so when you take a look at a graph, a voltage graph, so this is basically plotting your voltage over time of your action potential. So from a scale of minus 85 millivolts to plus 20 millivolts. So what you have here is your resting membrane potential. And eventually what's going to happen is you're going to get a depolarization event that's going to basically increase your voltage by a lot. So you see this sharp incline, this positive slope. This basically, it represents the depolarization, the point at which your sodium ion is rushing into the cell, flipping that charge. This is typically associated with the opening of those voltage-gated sodium channels. That usually happens right at threshold. Now then, you're going to get up to a certain peak where you're going to open up then your potassium channels. This allows for the reset. It's like a counterbalance to the inrush of sodium, right? Because you don't want sodium to rush completely inside. Otherwise, you're going to just completely blow the system. So what happens is the potassium gates open up. The potassium will flow out during the repolarization event, and this will basically balance out the sodium inrush. Because you don't want the sodium to rush in to completion. The potassium keeps the sodium basically from crashing. And so it will basically rush out as a counterbalance, as a counterforce to the sodium inrush. So basically with the potassium plus charge, the positive charge rushing out, it's going to dampen that depolarization, and you're going to get the repolarization where you get the reestablishing of your resting membrane potential eventually to normal levels. And this is when your sodium potassium pump kicks in and restores your resting membrane potential, which is right here. And so that's one firing, if you will, or one depolarization event of a neuron. That's what's happening in your membranes. Okay. So this is a good place to stop and a good place to start at the same time. So what we'll do is we'll take a look at this. So everything that I just covered is basically we'll review at the beginning here with these voltage gated channels and so forth, as we kind of take a look at sort of how this action potential initiation and then propagation works. So this is a kind of a good way to sort of go back over what we just talked about and to sort of review a little bit of that. And maybe hopefully this will be kind of like a second going over, and then maybe that'll kind of help it sort of stick a little bit more, so you'll sink a little bit deeper with the second
round of kind of going over it. Okay, so I will stop there.